much. And my name is Shaui. I'm now working at the University of Melbourne. And it's my great pleasure to introduce my project of investigating how menarche and menopause modify breast cancer risk. I completed my PhD in 2018, so I'm still still in my early career stage. I completed my PhD in University of Melbourne. After that, I went to University of Cambridge for a postdoc job working on breast cancer risk prediction. And I stayed in Cambridge for 2 years and came back last month. I just came out from the hotel quarantine, and luckily I'm still very well. And my research interests including cancer risk modeling, genetic and epigenetic epidemiology. A little background of my project. We know that early menarche and late menopause are associated with breast cancer risk. And these two events are thought to reflect the lifetime exposure to the ovarian hormones, especially estrogens. However, the precise mechanisms and pathways of how these two risk factors influence breast cancer risk are not that clear. And this project will investigate this question. And we will study demethylation. Demethylation is an epigenetic modification. It works in the DNA transcription process, and it will change how genes will be transcribed, but don't change the DNA sequencing. And our hypothesis is that menarche and menopause will change demethylation, then downstreamly change the protein production, and eventually change the breast cancer risk. So to test this hypothesis, we will do an epidemiological study. This is not like the previous talks. And we will use the data from 2 large population cohorts based in Melbourne. And these 2 cohorts have measured a massive data, including epigenomic, genomic, and epidemiological data. And this project will integrate these different types of data to test this hypothesis. And we will use modern causation assessment methods to try to find the pathways. And one of the methods to be used is called IceFalcon, the first one. This method is developed by us, and we have applied this method to many research questions and have shown that this method is very powerful in causal inference. A brief introduction of our project design. So for the women of the 2 cohorts, we have measured their demethylation levels at many sites. And here I only use the 7 sites as an example. At first, we would like to find which demethylation sites are associated with menarche or menopause. For example, we may find 5 sites are associated with these 2 risk factors, and the other 2 sites will not be investigated further. These 5 sites may be associated with these risk factors due to several reasons, like the first one, they may be due to confounders, or like the second one, they may be due to that the demethylation causes these risk factors. And our second aim is to find which of the sites are actually caused by these risk factors. So at last, we may find only 3 sites are caused by these risk factors. We will then further to investigate the relationships between these sites and breast cancer risk. There may also be several relationships. And our last aim is to find which demethylation sites cause breast cancer. And eventually, we will find this causation chain that menarche and menopause cause changes in demethylation, and demethylation causes changes in breast cancer. And through the mediating effect of demethylation, the menarche and menopause will cause breast cancer. And by studying which pathways the demethylation is involved in, we may find through which pathways menarche and menopause will cause breast cancer. And by finding these pathways, we can provide some evidence for the interventions to intervene the risk of menarche and menopause on breast cancer. And we are now doing this work, and our preliminary results are expected very soon. And at last, I would like to thank to my co-investigators. They have helped me a lot to my career, and we will work together on this project. And I also would like to thank support for the 2 cohorts and the funding I received from several funding agencies, especially for the Cure Cancer Foundation. Thank you very much.